Have you ever wondered how scientists measure gene expression? Well, it's a fascinating journey that delves deep into the heart of biology. Gene expression, simply put, is the process by which the information in a gene is synthesized to create a functional gene product, usually a protein. This process is vital in determining the biological blueprint of an organism. Now, understanding and quantifying gene expression is crucial for scientists. It's like reading the language of life, helping us unravel the mysteries of biological processes, from cell growth and differentiation to disease progression. There are several methods to measure gene expression, each with its own strengths and challenges. Some of these methods include northern blotting, quantitative polymerase, chain reaction, or qPCR, microarray, and RNA-seq. These techniques are the keys that unlock the secrets of our genes, transforming abstract genetic codes into tangible data. So how exactly do these methods work? Let's dive in. First off, we have northern blotting. This technique is a tried and true method to measure gene expression. It's all about separating RNA by gel electrophoresis. But what does that mean? Well, gel electrophoresis is a process that uses an electrical field to move molecules like RNA through a gel. Different molecules move at different speeds, allowing them to be separated from one another. Once the RNA is separated, it's transferred to a nitrocellulose membrane. This is a fancy way of saying that the RNA is moved onto a solid surface where it forms covalent bonds. This means the RNA sticks to the membrane, ready for the next step. Then comes the hybridization. This is where labeled probes, which are complementary to the RNA transcript we're interested in, come into play. These probes bind with the RNA, allowing us to identify and measure our target RNA. It's like fitting together pieces of a puzzle. Now, northern blotting has its pros and cons. On the one hand, it's simple and cost-effective. It doesn't require any fancy equipment or expensive reagents. On the downside, it's time-consuming and has low throughput. This means it's not the best choice if you're in a hurry or need to analyze a large number of samples. But remember, every method has its place. Despite its limitations, northern blotting is a reliable method for measuring gene expression. It stood the test of time and remains a valuable tool in the toolbox of genetic researchers. The next method in line is qPCR. The journey of gene expression measurement brings us to the shores of quantitative polymerase chain reaction, often referred to as qPCR. This method is a bit like a high-speed train, rapidly taking us from one point to another. It does this by converting mRNA into its doppelganger, complementary DNA or cDNA. Once we have the cDNA, it's time for amplification using the polymerase chain reaction, a process that multiplies the cDNA strands. It's like a cloning machine, creating replicas of the cDNA. However, the cleverness of qPCR doesn't stop there. Fluorescent labels are added, lighting up the process with every cycle. But why the light show? Well, this is how qPCR provides quantitative data. The fluorescence is measured after each cycle, and the intensity of the light corresponds to the amount of cDNA present. The beauty of qPCR lies in its simplicity and speed. It's relatively easy to use and boasts a short assay time, making it a popular choice amongst researchers. However, every method has its caveats. qPCR requires a prior knowledge of the target sequence, akin to knowing your destination before embarking on a journey. It also has a limited throughput, which means it can only process a certain amount of information at a time. Despite these nuances, qPCR is a quick and efficient method for measuring gene expression. It's like a reliable friend, always there to lend a helping hand when you need to navigate the intricate world of gene expression. Scene script. Next up, we have the microarray method. In the world of genetic research, the microarray method has established itself as a key player when it comes to measuring gene expression. So what is it all about? Simply put, the microarray method involves hybridizing complementary DNA or cDNA strands to quantify transcripts. Imagine it as a microscopic chessboard, where each square represents a different gene. Now, the beauty of this method lies in its ability to perform high-throughput analysis. In other words, it allows us to simultaneously examine the expression of thousands, if not tens of thousands of genes. This makes it an extremely efficient tool, saving both time and resources, which are often scarce in the realm of scientific research. But as with any other method, the microarray isn't without its drawbacks. It requires the separate preparation of control and test samples. This can be a time-consuming process and may introduce variability into your results. Additionally, the microarray method calls for specialized equipment for data analysis. This can be a barrier to entry for some researchers, as it requires a significant investment in terms of both money and training. Despite these challenges, the benefits of the microarray method are undeniable. Its ability to measure a large number of transcripts at once provides a comprehensive snapshot of gene expression, making it a valuable tool in the genetic researcher's arsenal. Microarray allows for the analysis of a large number of transcripts at once. Last but not least, we have RNA-seq, a method that's as intriguing as it sounds. RNA-seq is all about sequencing RNA and measuring expression by counting sequences. It's like taking a detailed snapshot of the RNA content at a given moment, providing us with a wealth of information about gene expression. The process starts with our RNA samples. These are then transformed into libraries ready for sequencing. Now imagine each RNA sample as a book in this library, each one holding its own unique story of gene expression. It's through sequencing these books that we unlock these stories. The data that emerges from this process is then analyzed to determine gene expression levels. And this is where RNA-seq truly shines. It provides a higher range than microarrays, meaning it can detect a wider variety of RNA types. So, whether you're dealing with messenger RNA, microRNA, or other non-coding RNA, RNA-seq has got you covered. However, with great power comes great responsibility, and in this case, that responsibility takes the form of higher costs and the need for more computational resources. It's true, RNA-seq isn't the most budget-friendly option out there. It also requires a hefty amount of computational power to analyze the massive amount of data it produces. But all things considered, RNA-seq provides a comprehensive view of the transcriptome, making it a powerful tool for gene expression measurement. So there you have it. We've journeyed through the fascinating world of measuring gene expression, exploring 4 compelling methods: northern blotting with its simplicity and cost-effectiveness, qPCR offering real-time data and ease of use, microarray enabling high-throughput analysis, and finally RNA-seq providing a broader range and the ability to detect various RNA types. Each method has its own strengths and weaknesses, but all contribute to our understanding of gene expression. Remember, it's not about finding the best method, but rather the most appropriate one for your research needs.